Good morning, dear students. Welcome to this new YouTube channel. In this YouTube channel, we will be covering science, social science, English, and French for the class 6, 7, 8th, and 9th. Currently, we are working on class 7. So, let's start. Class 7 Science Success by Goyal Brothers Prakashan. The first chapter of this science book is Nutrition in Plants. So what is nutrition? The process of obtaining the food is called nutrition. Okay. So process of obtaining the food is called nutrition. So what is food? Food is basically there are 6 components of the food: carbohydrate, fat, protein, vitamin, minerals, and roughage. These 6 component is called together called food. Okay, carbohydrate, fat, protein, vitamin, minerals, and roughage. And why food is required? Food is required for 3 things. What are those 3 things? Food is required for energy, food is required for the growth of the body, and food is required for the fight against disease. So nutrition is process of obtaining the food by the living organism is called nutrition. There are 2 modes of nutrition: autotrophic nutrition and heterotrophic nutrition. What is autotrophic nutrition? When living organism make their own food, such kind of nutrition is called autotrophic nutrition. How living organism can make their own food? They make their own food by the process called photosynthesis. Okay. When living organism are dependent on other living organism or other organism and they obtain the food from other organism, such kind of mode of nutrition is called heterotrophic nutrition, and the organism are called heterotrophs. Okay, so let us understand both of these type of nutrition in detail. For autotrophic nutrition is dependent upon the photosynthesis. So let us understand what is photosynthesis. So photosynthesis required, there are 4 components that is required by the plant. In green plants, they contain the chlorophyll. Okay, so chlorophyll containing green plant, in presence of sunlight, they take the carbon dioxide and water, and in presence of sunlight, so there are 4 components of the photosynthesis: carbon dioxide, water, chlorophyll, and sunlight. So in presence of these 4 components, green plant, by the process of photosynthesis, they synthesize glucose, and oxygen is released in the atmosphere additionally. Okay, so green plant in presence of sunlight and chlorophyll pigment, they take the carbon dioxide and water and synthesize glucose. This glucose is later on converted into starch. Okay, so this is the reaction of the photosynthesis. So we can find out the starch, presence of starch, by simple test called iodine test. Okay, if we pour the iodine solution in the starch, it will turn into blue color or dark blue, black color. So let us understand each of these 4 components in detail. First is chlorophyll. So chlorophyll basically help in absorption of sunlight. So first thing is what is the role of chlorophyll? Chlorophyll help in absorption of the sunlight by the plant. So it is a pigment. It is a pigment, and that pigment absorb the sunlight, and that energy of the sunlight is later on used for the photosynthesis. Okay, so this experiment we will study later on. Then second is what is cell. Cell is basically a basic unit of the life. Each organism consists of many cells, millions of cells, and structure of the cell is look like this. This is the cell. The outer membrane of the cell is called cell membrane, and each cell contains the nucleus inside. This is nucleus. Okay, and between the cell membrane and nucleus we have the cytoplasm. Okay, so this is the cell. Next component of the photosynthesis is carbon dioxide. So basically plant take the carbon dioxide from the atmosphere, but the carbon dioxide enter into the plant by pore, and this pore is called stomata. Okay, stomata is for plural and singular is called stoma. So structure of stomata is like this. Stomata consist of two guard cells. So these two are guard cells. Okay, so these two are the guard cells, and this is the outer thin wall of the guard cell, and inner side of the guard cell is thick wall. So inner side is thick and outer is thin. Okay, and guard cell is surrounded by many other cell which is called epidermal cell, and the pore is called stomatal pore. When it is open, we can see the pore, but when the stomata is closed, so pore is closed, and guard cell help in opening of stomata. Okay, when it is open, carbon dioxide can enter. Carbon dioxide can enter the plant. Okay, and now we can prove that carbon dioxide is required for the photosynthesis by this experiment. We will study this experiment later on. Next is sunlight. Okay, so sunlight is provided by the sun. Okay, sun releases the energy, electromagnetic radiation, and that energy is absorbed by the chlorophyll, and therefore sun is the source of ultimate energy, because sun gives the sunlight, and sunlight is used by the plant to form the food, and all the food on the planet earth is therefore indirectly is given by the sun. Okay, so that is why it is called sun is the ultimate source of energy. We can prove the role of sunlight by one experiment. We will study this experiment later on. Next is water. So water, root basically absorb the water from the soil, and it also absorbs some minerals from the soil. So water is also required for the photosynthesis. So these four components are required for the photosynthesis, and by the process of photosynthesis, plant make the glucose and oxygen is released, and glucose is later on converted into starch, and we can find out the presence of starch by the iodine test. So this is autotrophic nutrition. In autotrophic nutrition there is a photosynthesis, and in photosynthesis there is a chlorophyll, there is carbon dioxide, sunlight and water. Okay, so now heterotrophic nutrition. Heterotrophic nutrition means living organisms are getting food from other organism. Okay, so there are many plants also which do not synthesize their own food. They take the food from other organism. So there are four types of plants. First is parasitic plants. So any kind of parasitic means one organism take the food from other living organism. Okay, so other living organism is called host. Okay, and parasitic plant is taking the nutrition from other living organism. The other living organism is called host. So there are examples of such kind of plants. There is one plant called Cuscuta. Cuscuta have special tubular structure called hostoria, which penetrate into the host plant and take the food from the host plant. So Cuscuta, with the help of hostoria, take that food from the host plant. Other example is mistletoe, and third example is Rafflesia. So these are the three examples of parasitic plants: Cuscuta, mistletoe, and Rafflesia. Next is saprophytic plants. Saprophytic plants means when the plant take the food from dead organic matter. Okay, so there is no living organism as a host. It is the dead matter from which the nutrition is taken up. For example, mushroom. Okay, so mushroom, you can find the mushroom in the dead organic matter or simply matter. Okay, so we can prove growth of the fungi in an experiment. We will study the experiment separately. Then next type of heterotrophic nutrition is symbiotic relationship. In symbiotic relationship, basically there are two types of organisms which are helping each other. Okay, and example of such kind of symbiotic relationship is lichen. Okay, in lichen we have algae and fungus, both living with each other. Fungus provide the shelter, water and minerals to the algae, and algae by the process of photosynthesis synthesize the food, and this food is then provided to the fungus. So both algae and fungus are benefiting from each other. Similarly, legume plant also. In legume plants we can see rhizobium bacteria. So legume plant, in roots there are root nodules. Okay, this is the root and there are root nodules present. In root nodules we can see the rhizobium bacteria. So basically rhizobium bacteria have the ability to fix the atmospheric nitrogen, and this nitrogen is then provided to the legume plant, and legume plant provide the food in return of the nitrogen. So both are benefiting from each other. So in symbiotic relationship we have two examples, lichen and legume plant. Next is insectivorous plants. So in insectivorous plants there are different examples like pitcher plant, Venus flytrap and bladderwort. These all plant have ability to capture the insect. Okay, and these plants digest these insect, and they digest these insects and they take the nitrogenous nutrient from these plants, these insects, because these plants are growing in a soil where nitrogenous nutrient is absent. To fulfill their nitrogenous nutrient requirement, they capture the insect, they digest those insects by the help of digestive juices, and then nitrogenous nutrient is provided by the insect to these plants. So these are the three examples: pitcher plant, Venus flytrap and bladderwort. Okay, अब हम सीखेंगे ये जितने भी बॉक्स के अंदर आपके दे रखे हैं एक्सपेरिमेंट्स, उनको सीख लेते हैं. तो पहला एक्सपेरिमेंट है जिसमें ये बताया गया है कि क्लोरोफिल जो है वो आवश्यक है फॉर द फोटोसिंथेसिस. सो फॉर दिस एक्सपेरिमेंट हमें पहले प्लांट को प्रिपेयर करना होता है. तो इस स्टेप में यहाँ पे जो दे रखा है ये प्लांट की प्रिपरेशन कैसे करते हैं वो समझा रखा है. सो बेसिकली व्हाट वी डू, वी कीप द प्लांट in the dark room for 3 days. So in that 3 days all the starch is consumed by the plant, and then now the plant is known as de-starched plant. That means starch is not present in that plant. Then we keep the plant in sunlight for 6 hours. So during this 6 hour new starch is synthesized. Okay, so this is called preparation. So we have taken variegated leaf plant. Variegated means in such plant we have patchy area of chlorophyll, and some area where chlorophyll is not present. So those area are known as white area. Okay, so we after keeping the plant in sunlight for 6 hours, we pluck the leaf, we boil the leaf in boiling water for 2 minutes, then we keep the leaf in test tube having alcohol in this yellow color. You can see alcohol is there, and we warm this test tube in a water bath, not directly on the flame. We should keep the test tube in the water, and water should be boiling. Okay, so during this process all the decoloration happens, and then we take out the leaf. So everything is now decolor, only the green portion where chlorophyll is present, photosynthesis has happened. So now we pour the iodine solution. So after pouring the iodine solution, we can see that the area which were green in color, which means where the chlorophyll was present, due to chlorophyll presence, those areas synthesized the starch, and under the iodine solution, that starch is turned into blue black color. And those area which are white patches in which there is no chlorophyll is present, so there is no new starch is synthesized, and those area remain white color under the iodine solution treatment. So this experiment shows that chlorophyll is necessary for the photosynthesis. Next is carbon dioxide is necessary for the photosynthesis. So in this experiment, we have kept half leaf in a bottle, and in this bottle we have caustic potash solution. So caustic potash solution basically absorb all the carbon dioxide present in the atmosphere. So CO2 is no longer present in this bottle, and we keep the plant for 6 hours in the sunlight. So photosynthesis can occur in this area, but photosynthesis cannot occur in this area inside the bottle because there is no CO2. So if we take after 6 hours, we plug the leaf and we pour the iodine solution. As you can see in this diagram, half of the leaf turned blue black color because of the presence of starch under the presence of chlorophyll, carbon dioxide, sunlight and water. But in this half area there is no blue color because there is no starch. Why there is no starch? Because chlorophyll was because carbon dioxide was not present due to the caustic potash solution in the water. So this experiment shows that yes, carbon dioxide is essential for the photosynthesis. Then third experiment, this experiment shows that sunlight is required for the photosynthesis. So for this experiment, we can stick a tape on the leaf, and because of the sticky tape, there is no sunlight under this tape. So we boil the leaf in a water, and then we boil the leaf in alcohol for few minutes, and then we pour the iodine solution. And after iodine solution, you can see that under the tape, where there is no starch, the leaf remain white, while other area turned into blue black color due to synthesis of starch under the presence of sunlight. So there is no sunlight here, so there is no starch formation also here. So these three experiments basically gave the evidence that yes, chlorophyll is required, carbon dioxide is required, and sunlight is required for the photosynthesis. There is one more experiment mentioned here, that you can grow fungus in the bread. You take the bread and you keep the bread in water for 1 or 2 minutes, and then you place the bread in dark area where fungus started growing after 2-3 days, and you can see the brown or white patches showing the growth of the fungus. So in this experiment, it provide an evidence that yes, fungus is a saprophytic organism which take the nutrition from the dead matter. In this case, the dead matter is bread. and the fungus is growing on the bread after 2-3 days. So these are the four experiments. Thank you everyone.